Good morning, everyone, and thank you for being here today. I am pleased to welcome you to this presentation. Today, we will be discussing the topic of Dr. S.K. Singh's comparative dissolution profile. This is an important area in pharmaceutical analysis, and I look forward to sharing some valuable insights with you. Over the course of this presentation, we will explore the methodology, results, and implications of dissolution profile comparisons. So, let's get started and delve into the details. Let's start by talking about what dissolution testing actually evaluates. It measures how quickly and how much of a drug is released from a dosage form, like a tablet or capsule, into the body. This is critical because it influences how the drug works once administered. Understanding dissolution helps us determine whether two different formulations are bioequivalent, meaning they deliver the same amount of active ingredient into the bloodstream in the same timeframe. This is essential for ensuring consistent therapeutic effects. It also plays a key role in biowaivers, which are regulatory exemptions from needing in vivo bioequivalence studies, especially when formulations are based on the Biopharmaceutics Classification System, or BCS. Additionally, dissolution testing is important when making post-approval changes to a product, ensuring that modifications haven't affected its performance. Overall, these tests help us confirm that different products or changes still provide the same therapeutic benefit, maintaining patient safety and efficacy. Regulatory agencies often recommend comparing dissolution profiles statistically. This means using specific methods to objectively assess whether two profiles are similar, supporting decisions about equivalence. So, in the broader context of pharmaceutical development and regulation, dissolution testing is a fundamental tool to ensure that the medicines we use are safe, effective, and consistent. Now, let's take a closer look at the main objective of comparative dissolution. The goal here is to compare the test product, which we'll call T, with the reference product, R. Essentially, we want to see how they perform side by side in terms of their dissolution profiles. The key demonstration is that both products should show a similar release profile, meaning they dissolve at comparable rates over time. This similarity indicates that the test product will perform like the reference in the body, which is crucial for ensuring therapeutic equivalence. Another important point is to show consistent product performance. This consistency confirms that the manufacturing process reliably produces a product that behaves as expected, which is essential for both regulatory approval and patient safety. You'll notice that this comparison is used in several critical areas. It's vital for gaining approval for generic drugs, where demonstrating similarity to an established product is required. It's also important for SUPAC changes, those slight modifications in manufacturing processes, and for scale-up validation, where we need to ensure that larger production batches maintain the same performance as smaller ones. Overall, this slide underscores how comparative dissolution is a fundamental tool in ensuring that our generic and modified products meet the same high standards as the original reference. Let's take a look at the key acceptance criteria for similarity assessment. First, if the F2 value is 50 or greater, we consider the profiles to be similar. This means the test results are quite close in their dissolution profiles. Next, if the F1 value is 15 or less, that indicates an acceptable difference between the profiles. Essentially, a low F1 combined with a high F2 suggests the two products behave similarly. There's also an important point about dissolution. If 85% or more of the drug dissolves within 15 minutes, then we typically don't need to perform the F2 similarity test. This is because rapid dissolution often indicates similar performance. Now, looking at regulatory guidelines from the FDA, EMA, and WHO, they all recommend testing at least 12 units under the same conditions, at multiple time points. This ensures consistency and reliability in the comparison. The primary tool for this assessment is the similarity factor, or F2. It's a widely accepted statistical measure to compare dissolution profiles and determine similarity. Overall, These criteria and guidelines help us ensure our formulations are consistent and meet regulatory expectations. Now, let's take a closer look at the statistical comparison of dissolution profiles. Here, we're using various statistical tools to compare how different formulations dissolve over time. The key point is to understand whether these profiles are truly similar or significantly different, which can impact bioavailability and overall drug performance. We often use parameters like the similarity factor, F2, and dissimilarity factor, F1, to quantify this comparison. These factors help us make objective decisions about whether two dissolution profiles can be considered equivalent. Highlighting the importance of this comparison, it's not just about the numbers. It's about ensuring consistency in drug release, which ultimately affects patient outcomes. Connecting this to the overall theme, statistical analysis provides us with a robust, scientific way to validate our formulations and ensure quality control throughout development. It's a critical step in translating laboratory results into reliable, effective medicines. Let's take a look at the conditions for applying the F2 metric. First, it's important that the sampling times for both T and R are aligned, meaning they occur at the same time points. Additionally, we need at least 3 time points beyond 0 to get a reliable comparison. When it comes to the coefficient of variation, or %CV, we aim for less than or equal to 20% at early time points, which indicates acceptable variability early on. For later time points, the threshold becomes more stringent, with a %CV of less than or equal to 10%. Also, it's worth noting that if only one time point exceeds 85% CV, this can still be considered acceptable, but generally, maintaining these limits helps ensure the data's reliability. These conditions are crucial because they help us determine if the F2 comparison is valid and meaningful within the overall assessment framework. Let's take a look at this example of F1 calculation. Here, we're walking through the process step by step. The key point is understanding how the calculation works in practice, which helps us grasp the underlying concepts better. As we go through this example, notice how each component contributes to the final result. This is essential because it connects directly to the overall theme of applying theoretical formulas to real-world data. By breaking it down this way, you can see how the pieces fit together and how to approach similar calculations in your own work. Remember, mastering this example paves the way for understanding more complex scenarios down the line. All right, let's walk through step 1 of the F2 calculation example. The first step is to square the differences. What does that mean? Well, for each data point, we take the difference between the observed value and the predicted value, and then we multiply that difference by itself. This process turns all the differences into positive numbers and emphasizes larger errors. It's a crucial step because it helps us quantify the overall error in a way that's easy to compare and analyze. By squaring these differences, we're laying the foundation for calculating the total error, which will give us a clearer picture of how well our model is performing. So, remember, square each difference to move forward in our error analysis. Let's take a closer look at the interpretation of the F1 and F2 values. When we see an F1 value that is small, it indicates a minimal difference between the two formulations or samples we're comparing. Conversely, a high F2 value suggests a strong similarity. Now, focusing on the regulatory conclusion, if the F2 value is 50 or greater, we consider the similarity acceptable. This is crucial because such a result supports the case for a biowaiver or a bioequivalence claim, meaning that the test product can be approved without additional in vivo studies. This threshold helps streamline the approval process and ensures consistent quality standards. Remember, the key point here is that an F2 of 50 or above is a positive indicator, reinforcing the acceptance of similarity and supporting regulatory decisions. Now, let's talk about some limitations of the F2 method. First, it's quite sensitive to high variability in the data, which means if your measurements fluctuate a lot, the results might not be as reliable. Additionally, the F2 approach depends heavily on the number of time points you have. If you have too few, the method's effectiveness decreases. It's also important to recognize when this method isn't suitable. For example, if the coefficient of variation, or CV, is too high, the F2 method might give misleading results. Similarly, if your sampling times differ significantly between groups or conditions, it can compromise the analysis. So, what are the alternatives? When these limitations come into play, you might consider using bootstrap methods or multivariate approaches. These techniques can often handle variability and sampling differences more robustly, providing more reliable insights in complex situations. In conclusion, I want to emphasize that comparative dissolution remains a critical regulatory tool in our industry. It helps us ensure that generic products meet the necessary standards for approval. The F2 similarity factor continues to be the gold standard for assessing dissolution profile similarity. Of course, when using this method, it's essential to follow a strict experimental design and adhere to clear statistical criteria to ensure reliable results. These principles support important objectives like gaining generic approval and maintaining consistent quality across products. Overall, these tools and standards are vital for safeguarding patient health and ensuring the integrity of our pharmaceutical offerings. For further support, please contact us. We're here to help you with any questions or additional assistance you might need after this presentation. Remember, your success is our priority, so don't hesitate to reach out. Thank you very much for your time and attention today.